Today we're continuing to talk about other patterns of inheritance, and we're going to focus on multiple alleles, orders of dominance, and pleiotropies. Multiple alleles is exactly what it sounds like. It occurs when you have a particular gene that has more than just two different types of alleles. A good example of this is ABO blood type, the type that most people are most familiar with. The A type phenotype is controlled by an allele we call IA, B type by an allele called IB, and O type is controlled by little I. So the A and the B allele are co-dominant with each other. They can both express simultaneously, but the O type allele doesn't cause antigens to be produced on the blood cells, so it's recessive to the other two. So this is how we get our four different phenotypes for ABO blood type. If you are AB type, you have to have both the IA and IB allele. If you are O type, you have to have two little i's. You have to be homozygous for that. The AB, you have to be heterozygous for the two codominant traits. And then for A type and B type, you can either be homozygous dominant for the A and B respectively, or you can be a heterozygote between the A and the O, or between the B and the O. So there's actually two ways to get the A and O type blood. So that ends up giving us those different phenotypes. And just an important thing to note as well is just again, dominance and recessive does not mean something is more or less common. O-type blood in 2003 in North America was actually one of the most common types of blood. It's just that the reason that we always need more O-type blood is because O-type, you can only take O-type from another O-type. You can only accept that blood from another O-type donor. And even more specifically, when we bring rhesus factor into it, if you're O negative, you can only take blood from other O negatives. And while the O blood type is fairly common, O negative is much rarer and more difficult to come by. So now that we've mentioned rhesus factor as well, we might as well talk about that too. Rhesus factor is what makes your blood type positive or negative. If you have rhesus factor, you have positive type blood. If you don't have rhesus factor, you have negative type blood. So the dominant phenotype is the positive, which is just big R, and the recessive phenotype is the negative, which is little r. And this works on complete Mendelian dominance. But we can combine it with ABO blood type to make a dihybrid cross. So you can actually make really interesting dihybrid crosses with blood type because the two genes that you're working with, even though they both relate to the same thing, blood type, they work in completely different manners. So you can get very interesting patterns out of it. Some other types of inheritance patterns, you can have order of dominance. This is when you have multiple different alleles and you have one allele that is completely dominant and one allele that's completely recessive, but then the other ones fit somewhere in between. So a classic example of this is often with rabbit fur color. A brown fur color will be dominant to all other types. The pattern that we call chinchilla, which is kind of gray, is recessive to brown, but is dominant over albino and Himalayan. The Himalayan, which is white with some black spots on the ears, nose, and legs, is recessive to brown and chinchilla, but dominant to albino, and then the albino type is recessive to everything else. So you have lots of possible genotypes that can produce one phenotype because you have lots of different ways of recombining alleles. And you just have to pay attention to what's dominant to what else. Another inheritance pattern is pleiotropy. Pliotropy occurs the opposite way around, where you have one gene controlling multiple phenotypes. Good example of this is going once again to sickle cell anemia. In sickle cell anemia, the sickle cell trait controls the shape of the blood cell as well as malaria resistance and immunity. Those are two completely different phenotypes. They are related because both of them are controlled by hemoglobin shape, which is ultimately what the sickle cell trait is actually manipulating, but sometimes you do have these pleiotropies where you have one thing influencing multiple different traits in kind of a cascade. And finally, we also have some genes that are environmentally influenced. Sex in crocodiles is an excellent and famous example. Depending on temperature, an entire clutch of crocodiles will either be male or female, and it's not determined by their chromosomes. It's actually determined by the temperature at which they incubate. So the changing temperature is what activates or deactivates the genes that cause their bodies to differentiate in different ways. We also consider the environment important for heritability because environment can also determine non-genetic traits. Like if someone has tan skin, if your parents had tan skin and you live a similar lifestyle and live in a similar location, you will likely have tan skin as well. You can see this in a variety of different cases. So there is actually some heritability that's not genetic. We call this broad sense heritability. It takes into account both what's been passed down genetically and what is a consequence of lifestyle. And we contrast that with narrow sense heritability, which is the heritability we want to deal with mostly in Biology 30, and that is strictly genetic. What gets passed down through the genes as opposed to what is a consequence of living a similar lifestyle to your parents.